Welcome to the Ribosome Podcast for April 29th, 2021. I'm Luke Roberts, and today we're speaking with Dr. Alexandra Seletsky, a scientist at Arrakis Therapeutics, about her experiences.
Hi, Luke. I'm excited to be here to talk to you today.
Great. Thanks so much. So how are you? And the question we have to ask everybody, how's your life doing in this global crisis?
I'm overall doing great. I think I'm honestly very thankful that, I have a job, I'm happy, healthy, and that's all I can ask for these days in the craziness that is, 2021.
No, exactly. There's a lot of things to be thankful for, and most of them are small. But it's good to recognize them when we do get them. And so I guess one of the big questions, we'll get right into it, is how was it to work for someone who later turned out to be a Nobel Prize winner? I mean, can you maybe describe what an average week was like, and how did it compare to other labs? Was it like, did you walk in and go, oh yeah, this is different? Or was it just, did it seem kind of the same?
Yeah, so I guess maybe to ground this a little bit, we can start out with I was a rotation student in the Doudna lab in fall of 2012. So that's really kind of at the dawn of the CRISPR age, if you want to put it that way. The Cas9 paper that had come out of the Doudna lab had come out the summer beforehand, and things were just really picking up in the CRISPR world there. But for me as a rotation student walking into the lab, there was the Doudna lab was no different than any other lab. It was a well-funded lab, and other than that, I wouldn't say there was anything special about it in regard to like being a Nobel lab. It wasn't at the time. Yeah. It was just a well-funded, large, biochemistry lab that was doing some, exciting science that I thought was maybe something that I could work on.
No, that's great. And I think that probably is a bit of a relief. It is to people like me who go like, okay, it's not like you show up and you go, okay, this is clearly a different level, but obviously the well-fundedness is obviously played a big role, but it's, they're just like us, celebrities, like that whole thing, right? The labs, it's just like ours, but
yeah. And I think, maybe some of the aspects that have really enabled the Doudna lab to do well and be the powerhouse that it is over its now like 25-year span. It is not necessarily just the funding that helps. I think it's the people that Jennifer recruits, and just some of the infrastructure. I think Jennifer even acknowledged this in, I think, some of her Nobel Prize speeches and things, but she's had the same lab manager since day one.
Yeah, I saw that in an article. That's really cool.
It's really cool. Kai Hong is a powerhouse and like a whirlwind of activity. And, I always tell people like Kai Hong is the magic that makes that lab flow, because she knows where everything is in the freezers. She, makes all of our T7. She's like a whiz at transcribing RNA. So like she's kind of this institutional knowledge that sits in the lab that really, makes things go.
Yeah. And I think, a lot of that stuff really probably the behind the scenes people are the drivers, because, not to take away anything that, Dr. Doudna has done, she's done amazing work, but, she's not in the lab every day. She's got bigger fish to fry. And so without people like your lab manager, the research isn't getting done.
Yeah. I think the special touch that Jennifer has is that she's really good at seeing potential in people and potential in projects. And so, she's very good at helping you think through like what the impact of something's going to be, or how would you frame this to make it of broader interest and not just this niche little field that you've been, thinking way too deeply on,
right,
as a trainee in her lab.
Well, no, that's great. And so what did you work on in the Doudna lab?
Yeah, so I worked on a variety of things. So I honestly probably had the most impact on society that I will ever have as a rotation student, because I was part of the team with Martin Jennick, who was working on getting Cas9 to work in mammalian cells.
Wow.
So it was basically my, first steps into that lab, which seems crazy now.
Yeah, that's wild. God, in retrospect,
Yeah, exactly. Bright-eyed rotation student being told, like, why don't you try and get this to work? Like, you've worked in mammalian cells before. Like, well, let's just give this a whirl.
Wow. Yeah, that's incredible to think of, especially, with all the, the potential for therapeutics and everything down the road. It's, you can go back and look at your one lab book from that day and be like, here's the day I got it to work.
Yeah. So, I mean, I think that was just eye-opening. I don't know everything that was going through Jennifer's head when she was, pitching that project to a rotation student. But for me, as the, new person, really in some ways over-enthusiastic first-year grad student, like, it was an amazing project to be part of, and I'm really grateful for the opportunity. So then I went off, I did 2 other rotations at Berkeley, and then at the end of the year, decided to come back and join the Doudna lab. And I made either a very stupid decision or a smart decision, depending on who you talk to try and stay as far away from Cas9 as possible, because I didn't want to work on a field that was quite that hot and competitive.
Okay, yeah.
And so I spent the next 2 years of my time in grad school in the Doudna lab, working on an aspect of CRISPR biology called acquisition, which is basically within the CRISPR biology field. It's the way that a bacteria discovers and remembers a new target that it's going to want to target with the CRISPR system. So I worked on that for 2 years, admittedly had 0 success, and ended up switching projects at the end of my 3rd year. And so from there on, I was working on a project that was trying to target Cas9 to RNA. So how can we repurpose this great molecular machine to then have applications on RNA? Evolved into what became kind of my thesis project, which was working on the actual CRISPR enzyme that is the Cas9 for RNA, which is called Cas13. And so really the crux of what I worked on and graduated out of the lab on was this project doing kind of the biochemical characterization of Cas13, understanding what its various enzymatic activities could do, and turning that into an actual RNA detection assay.
Oh, that's great. And I think it's also nice every so often, as unfortunate it is to hear that, your first 2 years didn't really pan out. It's just sort of like everyone else, goes through those as well. You feel good. You feel better about your own sort of projects like that when you go, yeah, all right, good. Even, not everybody is, it's not just me. But I'm glad you managed, to switch and still get a very, productive PhD out of it. That's very good.
I think the other lesson learned is that you can look at my CV and see, these really cool, high impact papers. But the time in between those 2 papers was not so successful. So there was a lot of, challenge and failed projects and dead ends in between. So just because, it looks like there's been a lot of success or someone has that big paper, it doesn't mean that things were always working for them.
Yeah, absolutely. A lot of failures on the way to those successes. It's important to keep in mind, hard to conceptualize when you're just seeing the new papers come out every week in, in science and in nature, right? But you go, okay, for every one of these, there's like 100 projects that didn't get there.
Right. And I feel very lucky that I was on projects that, did have that potential to publish well, or that we were in kind of the right place at the right time working on science that, other people are going to be interested in, because that's not necessarily the case. So,
Yeah, sometimes just like you mentioned, sometimes you get a hot field. And if you're just fortunate enough to be in the right place, or you see it coming and you've got the tools to work at it, you can really cash in. But yeah, sometimes, as much of the stuff as you're doing right now, like if nobody's reading about it, cool as you think it is, it's hard to get it out there in those higher impact journals. And so I guess after your time in the Doudna lab, why did you choose to leave academia?
I would actually challenge you on that question because for me, it was not about leaving academia. It was about going back to industry. So to, again, maybe walk it back a little bit more, I had taken 3 years off in between undergrad and enrolling in grad school at UC Berkeley. And in those 3 years, I worked at Merck, which is a very big, large biopharmaceutical company, and then spent 2 years working at the Broad in one of their core groups that really functions a little more in an industry-like setting than an academic setting. And so, through those 3 years, I really figured out that I loved the team-based science that was going on in those groups. And I was part of, at the Broad, I was part of like an 8 to 10 person team that was pushing together this big project to screen 500 cancer cell lines by a genome-wide shRNA library. And so it was this huge part where everyone played a key role and we were passing things back and forth. And it was just really invigorating to me to be part of this larger team. And so I actually had a harder time when I transitioned into academia, going into this model where it was just you working on your project, trying to push your thing forward. And that was so foreign to me. So when I was finishing up, I knew that, my ultimate goal was to go back to that more team collaborative science as quickly as possible. So I was, lucky enough to land a job straight from my PhD.
I mean, that's great. And that's even, as somebody who hasn't had an experience like that, it's very, the only experiences I've relatively had are self-driven science. That's really interesting. I hadn't considered being part of a larger group and that being, more amenable to other work styles. But, good job challenging me on my question. If you know what you like, there's no reason to be in the academia ivory tower if, industry is your fit.
Exactly. And I think the other piece of it is that, I hope and wish that there was more conversations within academic settings, that there are other options of what you can do with a PhD, and that you don't just have to train as a grad student to go do a postdoc, to become a PI, to be on this track. And that's not to say that track works for some people and it's a great way to do science, right? But there are other options out there and it's not like one size fits all.
You said you were lucky to get a job out of your PhD. Would you have any advice for maybe some young PhD students who are looking to do just that? What should their next steps be sort of looking like?
Yeah, so I think as someone who's now sat on many hiring committees on the other side, the things that at least my company is looking for, and this is not necessarily every company, right? I can only speak for my own experiences, but the things that companies are looking for is for you to be productive on the job from day one. So generally that means that you have some skill set that they need.
Right.
And that can be really broad, right? That can be, an easy one to think about is having bioinformatics skills, right? And so, if you were someone who worked on a, very basic biology project, but handled a fair amount of sequencing data, like your ability to handle sequencing data is actually a pretty good marketable skill set within an industry setting. And we at Arrakis have hired, multiple people of that phenotype of kind of being a mixed wet lab computationalist for pure computational roles. Other ways to think about it is like when you are, approaching the end of your PhD, think about what your skill set is and try and find companies that care about that skill set. So for me, I was a protein RNA biochemist. Right? That doesn't necessarily in and of itself shout translational medicine.
Yeah.
At all. It's not like I did mouse work. It's not like I was a cancer biologist. I was a biochemist. So as I was searching for companies, I was looking for companies that cared about nucleic acids and really were going to be solving problems within their research organization that were going to be more molecular biophysics, biochemistry based. Mm-hmm. And so I think that's what landed me at Arrakis, which is Arrakis is a company that is trying to develop RNA targeted small molecules. So a lot of what Arrakis as a company thinks about is how do we connect RNA structure to function so that we can target it therapeutically.
Right.
So me as an RNA, protein biochemist fit pretty nicely into let's try and figure out RNA structure function questions.
And I think maybe one of the intimidating things about these larger companies is they've got a huge amount of staff and you go, well, I don't really know how to, when you say it right there, like, I don't know how to target small molecules. What could I be doing? And you go, well, that's not, you don't show up and they go, let us know when it's done. They go, we have very specific problems, that we're solving on like the fundamental level. And as long as you can like work through those problems, it's not like you mentioned by yourself and you don't have to wear all the hats. And
I think I got lucky that I'm at a company where there is good acknowledgement that, no one person is going to solve this. And so you're leveraging, expertise all the way from medicinal chemistry, because they're the ones who know a whole lot about how to make good small molecules, through computational biology, through RNA biochemistry.
I guess, do you think the CRISPR technology or gene editing in general is going to soon reach, humans, or is this something that we're still 10 to 20 years away from?
I feel like I should preface this by saying that I do very little CRISPR-Cas9 research anymore. I work for a small molecule drug company, and admittedly, I'm very happy sitting in the stands cheering all of the companies who are trying to develop CRISPR-based therapeutics. Yeah. So I just want to make sure people know I have no insider info here or like.
Yeah, don't buy stocks based on this, anyone. This is not advice.
Yeah. Exactly. I feel very privileged that something that I worked on in grad school at some point may be part of a therapy that goes into a human at one point, right? Mm-hmm. And I think that's super exciting, and I think that it would not have as much buzz and as much money being thrown around at it if there wasn't real potential there for CRISPR-Cas9 based therapies to be helping patients. CRISPR based therapies definitely are going to make it to market. I don't think anyone really doubts that at this point. I mean, if you look at the headlines, Vertex just gave $900 million to CRISPR Therapeutics to really
a small investment,
just teeny, to really push their hemophilia drug to market. I believe. I think I have all the details there correct. So there is definitely interest, and that's going to be impactful. But where honestly, I think that CRISPR-based technologies are, making a bigger difference is every day in the lab, right?
Oh, absolutely.
People listening to this podcast have used CRISPR-Cas9, in their work to do something. And so that to me is where the real power of the technology comes from. It is just doing those, basic science aspects of, okay, well, now you can actually make mutants. Now you can engineer cell lines so much easier and faster, in a way that like opens up fields of science in a way that wasn't possible or wasn't easy before. Not to say it wasn't possible, it just wasn't as easy. In the long run, where do I think CRISPR-based technologies are going to have the biggest impact is like actually in basic research. To me, that's where I think if you were, if I was a betting person, if you were to go, 20 years into the future and look at the therapies on the market, I bet there's some step in their development that involved CRISPR for basically everything.
You get so caught up in this, we'll cure every disease, we'll get rid of sickle cell anemia, we'll just, edit it out, all these things like that, right? And you go, But yeah, it's something I guess it's probably overlooked is just how much of an advantage this allows basic researchers, and how much, how much of every research idea is based on these basic principles, right? It's like, like the PCR, what that did for basic research. This is probably just going to be looked at as one of those, even, in terms of its huge change it's having on everything. Yeah, I guess, yeah, it's easy to get caught up in all the hype about, like the things getting injected into people, but the basic research thing is very important to keep in mind. So yeah, thank you. That was a good answer for that. Do you have any more advice for like junior researchers if they're trying to make a decision regarding how to like join a specific group? What was it about the Doudna group that you said, okay, this is the group I want to go with? And I guess which leads to following a project, even now in your time in industry? How do you know what do you think is some of the most important factors for going, I think this is like good, or this is a dead end and I need to know when to call it?
You had a lot of questions.
I know that was like 7. I can hit it again if you want.
I think the first of those that is easy to answer is like, what advice would I have for people when they're thinking about joining a lab or joining a company or making their next step? Mm-hmm. And I think for me, it's every step that I have taken in my path to where I am now, I've been always focused on trying to find a good fit. So where is a good fit for me personally, where I'm going to be in an environment where I can thrive? And I've honestly cared very little about what the actual science I was doing. I'm a curious person who likes to solve problems. I'll solve those science problems on just about anything. My undergrad research was on plants. I worked on actually viral vaccines at Merck. I worked on genome-wide shRNA screening at the Broad. I worked on CRISPR nucleases and microbiology for my PhD. And now I work on cancer biology, right? So like really broad set of fields. And the thing that has kept me engaged those whole times is finding environments where I could thrive. So having the people around me, whether that be my peers and probably more importantly, either my mentor or boss, be someone who I knew that I could work with, because that's what was actually going to impact my ability to succeed more so than like what precise science I was doing.
No, I think that's a great answer. As much as projects might be cool, if it's a toxic lab environment, you're going to end up hating it. And so...
Right. And one person, and what works for one person is not necessarily going to be a great environment for someone else, right? So, some labs are really deep and focused in one area, and the PI can be an extreme expert on this one system, and they're understanding every new facet of a project. And there are people who totally thrive in that environment, right? Because you get to ask really precise questions and dig into really precise mechanism. Mm-hmm. And I know maybe this is starting to become apparent after listening to me talk, but like I would get so bored in that situation, like working on like one thing on one topic, hearing every talk at lab meeting being on like a similar topic would have been mind-numbing to me. So like I knew going into grad school that I wanted variety. I wanted, a lab that was working on a diverse set of problems, maybe using a similar technology sort of thing. So that was just something that I looked for. But there are other people who don't necessarily want that.
No, I think that's great advice. What advice would you give people on, coping with their, we can say failures, but when stuff doesn't work, what advice would you, like, how do you personally deal with it? And do you find certain strategies allow you to sort of like move on a little easier than others? What have you sort of found works for you?
Failure is part of being a scientist. So one, accepting that not everything succeeds. And if every experiment you're doing is working, you're probably not doing the right experiments, right? Because you should be pushing the boundaries of what is known or unknown. And a bad experiment is really only an uncontrolled experiment. The other, I think, key thing is ask for help. Ask the people around you if they've encountered these sorts of problems before, or like, is there a better control you could be running? Or I have this perplexing data. I don't know what to do. Like, what would you do next? Because everyone loves to talk and thinks that their ideas are the best. So you're like not actually putting someone out by asking for their help. Most people want to help you. Don't be afraid to ask for help.
I think that's really good advice, especially on the grad student type level when you're pushing your own project forward. It's very easy to get caught up in your own ideas and think like, I just need to do this again, right? And sometimes getting that outside perspective is super critical.
Or thinking that you have to be the one with all the answers.
Yeah.
Like, you don't have to be the person who knows everything, who designed every bit of your project, right? Some of the most fruitful things I did during my time in the Doudna lab were because I was talking to other people and they were, throwing out wild and crazy ideas at me. And we tried some of them. A lot of them didn't work, but some of them did.
All right. So I guess, since we just talked about coping and dealing with failure, do you have a particular failure of your own, which you'd be comfortable sharing that, this is one thing that we like to do at the end of our podcast is just, talk about, as much as we love to celebrate the successes, it's also, I think, equally important to, talk about the failures and, just even, if they're particularly funny, that's always a bonus for me.
I admit I don't think the story that I have is that funny, but I think it's very relatable, which is that I kind of already alluded to this earlier in the conversation, but probably the biggest, scariest failure I had in grad school was when I realized that I really needed to switch projects because I had been going down dead ends for 2 years and there was, nowhere else for me to go with this project. The project that I had was the biggest failure was, I mentioned it already, was the first project I was working on in the Doudna lab after I joined the lab full time. I was working on aspects of CRISPR biology that very few other people in the Doudna lab were working on. So I was really isolated. Mm-hmm. I was trying to do basically a microbiology project in a biochemistry lab, which was a poor choice in retrospect. But, I was early grad student who had big ambitions and wanted to drive my science in the direction that I thought was really cool. So I spent 2 years developing and working on this project. It's what I proposed for my qualifying exam. And 2 years in, I had to admit to myself that nothing was working. I don't think I had a single piece of positive data. To kind of add insult to injury, this was a period of time where my mentor, Jennifer Doudna, at the time did not have an administrative admin, which is kind of crazy.
Wow.
So somehow meeting with me had totally fallen off of her calendar. As I was realizing I needed a new project, I had not met with Jennifer for probably 7 months. So she had no idea that I had dug myself this huge hole. And I had to schedule a meeting and walk into her office and say, I haven't talked to you in 7 months. And I need a whole lot of help and a whole new project because I'm not going anywhere.
It takes a tremendous amount of courage, but I think obviously you made the right decision. I think that's really important, especially for some grad students to hear that might be in a similar position, right? Admitting to yourself that this isn't working and I can either bang my head against the wall and be extremely unhappy, or as hard as it is, making a change might be the right call.
Well, and I also think I had built up that meeting in my head because I was like, I haven't met with her. I have to have some piece of data. Like the next piece of data, that's when I'll schedule the meeting. The next piece of data, that's when I'll schedule the meeting. And it had just, snowballed into this, very, the situation that no one wants to be in, right?
Yeah. I've been there myself. Yeah, I've done the same thing. We go, I don't want to talk yet. I want to give me one thing, and then I can just have this one thing. Yeah,
one experiment that it worked. But I mean, I also have to, give Jennifer a ton of credit in that moment that she was 100% understanding, and so she was just like, "This is fine. This is normal. We can work through this. Stop trying to do experiments." And let's figure out something else for you to work on. I don't think all PIs would have said that in that moment. So, definitely it goes to her credit that she knew how to deal with me in that moment. But I think the other thing that she was doing was giving me space and not putting more pressure on me to like, oh, well, let's just do a few more things here. She was like accepting of my conclusion that this was not going anywhere and like, let's find something new. And so she pointed me in the direction of a few other postdocs. And that's where I really just started like realizing I need to be talking to people. I need to be asking for more help. I need to be, working on something that's not off in this corner, but actually like within the bread and butter of the lab of biochemistry. And that's where I'll actually have more success.
Well, no, that's really great. And, that's the interaction with a PI that you always wish you would have. So it really speaks to her, her character, and her ability to run a successful lab is probably due in large part to how well she handles these situations. That's really good to hear. I'm glad it went so well. And so I guess now that we've talked about your time in the Doudna lab, your time at the Broad Institute, and your current job now at Arrakis, what's next for you? Do you have plans, goals? I imagine you've definitely got them. I guess, just what are they?
No, I actually, when I saw this question on the question list of what you wanted to ask, I was like, I don't know what my answer is. I haven't thought about it. I think part of it is like once, in an industry setting, there's kind of less of like, here's your benchmark, right? Like, it's not like I'm aiming to start my own lab and or get the next, R01 funding or whatever. For me, I'm at the moment very happy with my current position. I get to work on a wide variety of science. I'm doing things from NGS assay development to cell line assay, basically validation. And what the next step for me would just to find a role that maybe had a little more visibility or more, influence across the org. But personally, I'm very happy and content where I am because I get to do cool science with cool people in an environment where I get to do fun things.
And I think that's a perfectly acceptable answer. I think a lot of people get tied up in the, there must be continual progress. I must get a promotion. I have to keep moving up the ladder. I got to do this. I got to do Y. And I think just, recognizing that you're happy in your job, you're thriving. There's, if your plans for right now are to enjoy your life, I don't see how that's a bad plan. So, and I think maybe that's a lesson some people can take home as well as, there's nothing wrong with just, you don't have to have a major plan.
So, I think the other piece is, I'm happy right now, but that doesn't mean I don't have ambitions or, there aren't places, where I want to, move up it within, the corporate ladder, although that sounds weird. For me, what, as long as I'm doing good science, I'm happy. So, I'll slowly develop as a research manager within industry and manage either larger teams or bigger projects. I don't envy the people who want to jump straight from a postdoc to a PI because those are two very different jobs. And while there are PI-like roles in industry, you move to them a little bit more slowly and stepwise in a way that's a little more comfortable for me personally.
I think that's great. And I think that's a perfect place to sort of wrap things up. And so I'm going to thank you so much for your time, Dr. Seletsky. And so just so everybody is aware again, Dr. Alexandra Seletsky is a scientist at Arrakis Therapeutics. You can find, do you have a social media you'd like to plug or something? Do you have a Twitter? Do you just want people to add you on LinkedIn? Do you want people to leave you alone? What would you prefer?
I am on Twitter at Alex Seletsky. I can't say I'm very active, but people are welcome to follow or reach out to me there. I would always plug that Arrakis is a great place to work. So if anyone has any questions about that, you can reach out to me on Twitter or LinkedIn. And we're always looking for, good RNA biologists.
All right. Well, hopefully there's a number of them listening right now, and you'll get an influx of junior scientists applying to this. And so if you're interested in more about the Ribosome Podcast, you can find us on Twitter at the Ribosome Podcast. And so this sort of concludes our episode. If you have comments or suggestions for the show, you can write us at theribosomepodcast@gmail.com. The show was edited and produced by Liana Boris and the rest of the junior scientists. And thank you so much for listening. Keep your bench clean and your RNA pure.